That's got it. Well, good afternoon, everyone, and welcome to this CWRH seminar. This is one that we've been trying to get in the books for some time now, and I am so delighted to introduce Dr. Michal A Elovitz, who is going to be our speaker today. Dr. Elovitz is the Hilarie L. Morgan and Michal L. Morgan President's Distinguished Professor in Women's Health at the University of Pennsylvania School of Medicine. And we collaborate with them on just, as a department and as a center on many different levels. She is a tenured professor there and has created one of the most successful maternal and child health research centers in the country. And we saw that with CHAP, where we had a tremendous contribution from our Penn partners. Dr. Elovitz graduated from the University of Texas and the Albert Einstein College of Medicine. She completed her residency in obstetrics and gynecology at Cornell University and her fellowship in maternal fetal medicine at the University of Chicago. I've known and I've been aware of Dr. Elovitz for over 20 years, and during that time, the association has been with Penn, where she's had multiple leadership roles, both there as well as nationally and internationally. She's very well recognized for cutting edge work in maternal, reproductive, and child health from the bench to the bedside. And I think there are very few people who cover those areas as much. And she's a strong advocate for women's health, really strong advocate for women's health, and just a fantastic mentor. I'm very, very delighted to hear Dr. Elovitz speak today on the microbiome and many other aspects that affect women's health and infants' health. So thank you. Michal, over to you.
Thank you, Alan, for that generous introduction. I can see in the conference room. Can you guys nod the front row who's eating? I really want food, by the way. And can you hear me okay and see me okay? Yeah? Okay, awesome. It's the one thing, so I love that I can, for you guys who don't know, Birmingham is actually, I grew up there. And so I would have loved to be in person. I think there's benefits of Zoom that I get to be with you guys, but I also wish that I could be in person. But regardless, I'm going to talk to you today about our work in the cervicovaginal microbiome. You will hear me call it CV microbiome. I don't really think it's just the vaginal microbiome. When I say CV, I don't mean cardiovascular health or the cardiovascular microbiome. So hopefully you'll get used to that. I'm happy to discuss more at the end. If there's questions, put in the chat and I can get through them at the end. I don't see that you guys in the conference room have computers. So at the end, just raise your hand or however it works there, and I'll be glad to talk about it. So just my disclosures are here, salary support from 3 different institutes at NIH, as Alan mentioned, and I'd love to talk to anyone about it. Very big advocate for women's health and women leadership. And so I co-founded and direct the Women's Health Collaborative. I'm a consultant. You can find my lab on that page. And for those who know me, you know I'm pretty active on Twitter. You can find me there. So on to the science. As I don't really need to tell a room of obstetricians, though I do see other people on the call, preterm birth remains a humongous burden for patients, their families, and for the children themselves. Occurring 1 out of every 10 pregnancies, probably in Alabama, that's about 1 in 9. And we really have I've been doing this, as Alan said, actually longer than him, about 25 years. We've made really minimal advances in preventing preterm birth. So that has been a driver for a lot of my research. The way that I've attacked understanding preterm birth is really this 3-prong approach. For those of you thinking about doing translational research, this is kind of the paradigm I've set up over the last 2 decades to really understand the drivers of adverse outcome and how we may best attend to them with available or novel therapeutics. So when we think about the current paradigm of preterm birth, and this is a schematic from a review paper I wrote in 2004. So 18 years ago, and I will tell you, I believe this as well. This is a few years after I finished my fellowship and I This was something I'd used in my first grant in actually 2001. So the current paradigm is right that we have microbes that sit in the cervicovaginal space. They ascend through the endocervical canal. They colonize the decidua. They replicate in the placenta. They cross over to the fetus, either hematologically or through breaking down those fetal membranes. That then triggers a bunch of different signal transduction pathways that lead to myometrial contractility. That myometrial contractility then begets cervical remodeling, and hence we have preterm birth. So based on that paradigm, right, that I and so many of my colleagues in this field believed, if it's uterine contractility leading to spontaneous preterm birth, then if we do clinical trials targeting myometrial activity, we should definitely prevent it, right? So we did that. Many of them done at UAB. We did a lot of clinical trials costing billions of dollars. The outcome of all those trials is no benefit and no change in the preterm birth rates. Okay, so maybe it's not myometrial contractility, but that paradigm is still correct. Let's look about uterine infection. So again, one of the trials I love at UAB where they did endometrial biopsies in between pregnancies, treated with antibiotics in an attempt to prevent preterm birth. That, along with so many other trials from the MFMU network and others looking at atypical bacteria, bacterial vaginosis, all of these trials with the goal that if we prevent uterine infection, we will in fact prevent preterm birth. And the outcomes of these trials, same thing. No benefit, no change in the preterm birth rate. So why did this paradigm fail? So a few things. The idea that microbes live in a cervicovaginal space, which is this really potent epithelial mucosal barrier, without interacting, and that barrier is inert, is just not biologically plausible. It was a total miss in our field. Most cervicovaginal microbes are actually non-motile, so they don't easily go back between the lower reproductive tract and the upper reproductive tract. The cervix is a biomechanical tissue unlike anything else in the body that responds both to load and stretch. The idea that the immune response in the CV space is this pro or anti-inflammatory response is really a reductionist approach that we really need to stop in OBGYN. It is much more complex than pro or anti-inflammatory. So about a decade ago, since I admitted that I had that same paradigm and is how I framed my first grant, I really started, as so much of what we tried to translate to the bedside failed, really try to conceptualize a different model that may lead to preterm birth. And we used what had been discovered outside of OBGYN and how that might apply to our world. And so really, what we really started beginning to think about in my research program was the cervical vaginal space is really its own unique biological niche. And in that biological niche, you have different epithelial barriers, you have the microbiota, you have the metabolome, you have biomechanics of the cervix, transcriptome of the epithelial cells and the immune cells, and really this entire ecosystem occurring in this unique biological niche. And the way we thought about it was this. So there's this epithelial barrier that's much more complex than this. In the vagina, the endocervix and ectocervix have three different kinds of epithelium. In the cervix, there's this mucus that protects the epithelial barrier, very similar to the gut, and that there's these microbial communities that live in the space. They induce at baseline this somewhat of an immune response that then the underlying tissue under that epithelial barrier, really unique to the CV space, is the cervix, which has fibroblasts, collagen, and extracellular matrix that holds it together, allowing the fetus to stay intact and to prevent parturition. So we think this is a model of reproductive health. And what we theorized was that when you have a disruption in those microbial communities, that the mucus gets compromised, the epithelial barrier gets compromised, you get this really potent diverse immune response leading to degradation of extracellular matrix, cervical remodeling, and that in fact is the primary event leading to preterm birth. So this was our hypothesis. We needed to prove it. So first, let's just focus on the microbiota. So the Human Microbiome Project really advanced our understanding of microbial communities across different body sites. This is one of the covers that I love after the Human Microbiome Project. So you can see here, they talk about different species at all these different body niches. This is a female noted by her mammary glands. I would bring your eye down to here where it says 60 species in the urogenital tract. And it talks about ureaplasma and a few others. You'll notice there is something missing on this female. She doesn't have a uterus or vagina. And we know that the uterus and vagina actually have their own microbial communities and are very critical to reproductive health. So when we thought about this, when you think about this, the cervicovaginal space in the lower and upper reproductive tract, we know that the cervicovaginal microbial communities exist. We know that they're there. So the question is, what are they? So my collaborator, Jacques Ravel, did this really seminal work in the late 1990s, where he took cohorts of non-pregnant women and looked at the microbial communities that existed over a reproductive course. And this is a heat map on the right side of your slide. And what this did is it laid out the different microbial communities that are normally there, if you will, in reproductive age women. And so I'm going to draw your eye to this line, and hopefully you can see this. You can see this heat, this intensity right here. So this is what's called community state type 1, and it's Lactobacillus crispatus. So lactobacillus is something we routinely associate with reproductive health, with vaginal health. And so a number of individuals in this reproductive age cohort were colonized with this what's called community state type 1. In the complete opposite of that, if you can see this really purple and pink area on the heat map, these are all anaerobic communities that make up community type state 4. So Ravel and his colleagues really came up with this community state type classification, 1, 2, 3, and 4. And for the purpose of this talk, we're going to talk about 1 being crispatus, which is really this healthy microbiota, and 4, which is a lactobacillus deficient state, common with Synethii, Mobiluncus, and Gardnerella vaginalis. That is community state type 4, and is believed to be associated with adverse reproductive outcomes. So this to me was very important in his paper in the late 2011, I said 1990s, I apologize, to look at, these are individual participants across 4 months of time, right? So these are weeks down here. So I just want to draw your eye to how this changes over time. So if you look at this individual who's in table A, the orange color is Lactobacillus inners, which would be community state type 3. So what's very interesting is that she shifts around week 9 to a non-lactobacillus dominated community, or CST4, filled with anaerobes. And what was so interesting about the study is the participants tracked vaginal symptoms, intercourse, menses, everything. And so what we know is that this individual shifted to this community state type 4, or to anaerobic. So what's very interesting that Jacques did here is they actually tried to assess these strains that grew up with the assumption that they must have been really low lying up here. Can you guys see my arrow? What's wrong?
Sorry, please go ahead.
Yeah, it's okay.
Yes.
So because the front row that I can see there, can you guys see my pointer or no? Yes, okay. So up here, you can see these little blue areas that were anaerobes. So the idea was that these anaerobes were in the cervicovaginal space, and due to something about sexual intercourse, were able to grow up. What I can tell you years later that we know is that that's actually not true. These strains of bacteria that were found in this individual at this time were actually transferred from sexual intercourse from the penile microbiome. So just keep that in mind when we think about STIs and BV and other things. Not as much for this talk, but for other discussions. In this individual, they were dominated in CST1 by Lactobacillus crispatus, optimal reproductive health, with these changed to inners during, I don't know if you probably can't see this, every month. So menses in this individual was associated with a shift in the microbiota. On the bottom, you can see what looks like a very kind of disordered microbial community. This is CST4, or lactobacillus deficient. So when we talk about CST4 and anaerobes, we usually think in OBGYN about bacterial vaginosis, and we think of it as an adverse outcome. But I will tell you what Jacques found in this study, there were numerous individuals that had CST4 that had no symptoms of BV. So we need to kind of rethink or reframe that CST4 or lactobacillus deficient may be a reproductive risk, but it not in itself may be associated with an actual disease outcome. So this was all in non-pregnant, and prior to this work in 2000 that we published in 2019, all the pregnancy data, not uncommon for our field, had cohorts of about 20 to 40 people. You cannot appropriately do the rigorous research that you need to understand the vaginal microbial communities with those small sample size. So with thankful funding from NINR, we undertook this really large cohort study of 2,000 individuals, and we called it the Motherhood and Microbiome Study, or M&M, and we published this in Nature and Communication, our results in 2019. But basically, we collected cervical vaginal samples at these 3 times in pregnancy, We adjudicated all the outcomes of the 2,000 individuals for medically indicated spontaneous preterm birth or other obstetrical outcomes. So pertinent to this talk, what we first found, so these are the different CSTs. So this is CST1 that I talked about, Lactobacillus crispata dominated, and here is CST4. It's actually more, this is an A and B, non-lactobacillus dominated. So the first thing that we found that was really interesting to us is that if you look at, so this is individuals who's reported as a Black race, and these are reported as non-Black, or for our cohort, were reported as white. And you can see here that there's this really big difference early in pregnancy about how many people were colonized with what we deem to be an optimal microbiota. So it's 45% of 50% of white individuals had this lactobacillus rich community, where only 22% of Black individuals did. If you reverse it and look at the non-lactobacillus dominated, and the numbers are up here, it's 45% of Black individuals early in pregnancy had the CST4 or this non-lactobacillus dominated community, where only 12% of white individuals did. The real question we had for the study, so that was unexpected finding, no one had ever reported it, but the real point of the study was, did they differ by race? By preterm birth status. So we did a case control from our 2,000 women. We looked at 432 term deliveries and 107 cases of adjudicated spontaneous preterm birth at less than 37 weeks. And what we found was about a twofold increase in spontaneous preterm birth in individuals who at 16 weeks of pregnancy were colonized with a non-lactobacillus dominated microbiota. We then did phylotype analysis because of having this many number of women enrolled, that we really could say what were the microbes really associated with preterm birth. So this is a volcano plot. These are your significant, it's a log Q value. This is the effect size on the bottom. And what I want to point out is that we really saw, which had been seen in some BV and HIV work, this really high effect from Mobiluncus mularis, which is an anaerobic, dissimilar from Gardnerella vaginalis, but really still associated with adverse outcomes. And we found this really high effect size with Mobiluncus and spontaneous preterm birth among all our participants, as well as those that identified as being part of the Black race. So then what we got really interested in Mobiluncus was, as this really kind of potent anaerobe, did the amount of abundance of that anaerobe in the CV space dictate the risk of preterm birth? In a lot of the preterm birth literature, as I think many of you know, we really talk about risk. It ends up being looked at for late preterm birth. But what we know the greatest burden is early preterm birth. So if you look at this Kaplan-Meier survival curve, the ones in blue had below the mean abundance of Mobiluncus. So they had less Mobiluncus than these individuals. And as you can see, individuals with a high load of bacterial abundance of Mobiluncus had a significantly increased risk of spontaneous preterm birth. And if you can see where the curves separate, it's really at this early gestational ages between 28 and 32 weeks. So we can begin to think about how an increasing bacterial abundance of specific anaerobes may increase the risk of preterm birth. So, but we have to keep balance, right? These good microbes and these bad microbes, and they live together in a community. The cervicovaginal space is not a monoculture, right? There's many different microbes living there. So how do they interact together? So this is showing you what I just showed you on the last slide, is that as we increase relative abundance of Mobiluncus, as seen on the x-axis, the probability of preterm birth goes up to where the highest levels, women had a 60% rate of preterm birth. And again, this is in 377 women. So we looked at the amount of good bug of lactobacillus decreased or mitigated that risk. So we divided all lactobacillus into tertiles. So tertile 1 was the lowest level. Tertile 3 is obviously the highest level. So if you look at individuals who had high levels of Mobiluncus and low levels of lacto, you can see that we had no impact on the probability of spontaneous preterm birth. When we get to the medium level of lactobacillus, you can see it flattens. That high level of Mobiluncus now is not associated with a preterm birth rate, with a high preterm birth rate. Same thing when we see the higher quartile. So we really see this interaction between classically good and bad bugs and how they interact in the CV space. So the question is though, and initially as a Bentz researcher, this is interesting. Lactobacillus deficient communities are associated with preterm birth. Specific anaerobes are associated with preterm birth, but as we must learn in our field, association does not mean causation. So how do we know these bugs are mechanistically involved in causing preterm birth? So we didn't. And this is what we've been working on for, again, the last decade. So this is kind of the prior paradigm that I showed you. And this is our evolved paradigm. And again, this is what we sought out to prove using in vitro and in vivo models. Do these different steps happen? And are they mechanistically involved in changing the epithelial barrier and ultimately cervical remodeling? So it's really important, and I think this gets lost in individuals that are outside of our field, that the cervix and the vagina evolves or is derived from two different embryological origins, right? If you remember back in embryology, the lower 1/3 and the upper 2/3 and the sinovaginal bulb and how everything evolves, there are different epithelia in the CV space. So we kind of conceptualize it like this, as you can see in blue, right? This more layered epithelium in the vagina, the transitional zone, and then we have columnar cells up in the ectocervix. And so what we have done, and we thought it was really important for our work, was to look at these different epithelial cell lines. And so we use vaginal epithelial, ectocervix, and endocervical cell lines to test our hypotheses. We also use different kind of bacteria. And again, this is a really reductionist approach. As I just told you, there's really vaginal microbial communities, but we've reduced this so that we can begin to look and tease apart mechanism. So we use Lactobacillus crispatus as our quote, healthy microbe, Gardnerella vaginalis, because there's just been so much about this anaerobe and its association with bacterial vaginosis, STI acquisition, infertility, and really a lot of adverse reproductive outcomes. And then we were really interested in studying Mobiluncus because of what we just showed you in the motherhood and microbiome cohort, that this was the microbe most highly associated with spontaneous preterm birth. So I'm not going to walk you through. These are our three most recent papers over the last few years that really started to dive down into how these microbes disrupt the epithelial barrier, the specific molecular mechanisms responsible for it, and how that will drive the ultimate of cervical remodeling and preterm birth. So I'm just going to walk you through a bit, but these are the three references for the data that I'm going to show you. And this paper to your right of your slide, we just published about a month ago in Microbiome. So first, the first question is, right? So I can't hear you guys to say this, but when you diagnose BV, what do you look for? Just nod your head if you pretend you know, you can pretend to answer me. Right? We look for clue cells, right? Okay, thank you. I see someone nodding their head. I appreciate that. So when we culture these, we want to be able to recapitulate what happens in human pregnancy or in reproductive age. So we wanted to look at how they look in our cells. So these are Lactobacillus crispatus floating around epithelial cells, and you can see they don't really adhere to the epithelium. They kind of float around. They seem fairly happy. They can live like this for quite a while. And then here's a picture of GVAG. I hope you can appreciate this. I don't know how it's projecting, but these are clue cells. They're just clue cells in my lab. And so these G. vaginalis really stick on the epithelial cells and adhere to them and really almost take over them. So that made me feel good in that we were studying something that appeared to recapitulate what we see clinically. But then we want to ask the question, if they're sticking to these cells, are they calling cell death? So this is a graph, it's LDH, was a measure of cell death. For this purposes, I'm showing you our work in ectocervical cells. And we took Lactobacillus crispatus, again, in the dark green, and in the hatched is G. vaginalis. And we did increasing dose on the epithelial cells. So you can see when we do 10 to the 6, we have a high rate of cell death in the ectocervical cells. And we still see cell death even when we go down to the 10 to the 4th. You'll notice that no matter how much lacto we put on these cells, the cells do not die. We wanted our hypothesis, as you remember, hopefully from my graphic, is that the epithelial cells are actually compromised by these non-optimal microbes, that the anaerobes disrupt that epithelial barrier, leading it to be more permeable, thus letting the influx of all different cytokines, immune cells, and everything traffic back and forth between the CV space and the cervix. So this is just an example of what we saw. This is cell permeability. So the higher the fluorescence is, the more permeable the epithelial barrier is, the less intact it is. So again, you see that with crispatus, it's almost inert on these cells. Nothing happens, where live GVAG really compromises the epithelial barrier. So then we want to know about the immune response. And as I noted in the beginning of the talk, the idea that there is a reductionist good and bad inflammatory response in the CV space is just way too reductionist. So this is from a Luminex array. I'm not going to walk you all the way through this, but what I'm going to tell you, this top row is the fold change. And so the darker red is the higher fold increase in the cytokine from L. crispatus on ectocervical cells. And this is G. vaginalis on ectocervical cells. And the same thing. And you can see here, right? So this is GCSF, one of the, quote, pro-inflammatory cytokines. It's 300-fold increase from putting a GVAG on the epithelial cells. But Crispatus increases at 40-fold increase. So this idea that I just showed you that Crispatus is sitting on the epithelial cells and is completely inert is a little mystifying, because this good microbe is still having an immune response. We do see a more detrimental and a more potent immune response with GVAG. But what I would argue is that the immune response in the CV space in these different epithelial barriers is quite complex. And to really figure out how this leads to adverse reproductive outcomes, we're going to have to understand this more. But what this work told us, and this is again in this paper down here that was published about 2 years ago, is there very much is a microbe-specific effect. And there are divergent immune responses between optimal, which is Crispatus here, non-optimal microbes. So if you remember, I showed you our human data where lactobacillus decreases the risk of mobiluncus. Okay, so we're seeing that again. Is association true? Can we actually see that in vitro? Can a good microbe limit the effect of a non-optimal microbe? So what you see here is, again, we're back to cell permeability. So this is our control, right? And then these are all the different lactose strains that we see here, and none of them change the cell permeability. What you see here is G. vaginalis. And again, we have this really potent effect where it disrupts the epithelial barrier. So then what we did is we gave G. vaginalis, but then we gave back all different strains of lacto to see if it prevented this breakdown of the epithelial barrier. And it does. So it doesn't completely get rid of it, but it definitely mitigates it. So hopefully in your mind, you're thinking, if lacto can mitigate the effect of GVAG, have we missed the idea of what therapeutics would be using to optimize reproductive health? And that would be a really good thought if you thought that. But then we got really curious about how live bacteria versus the products it makes that it spills out in the CV space, right? So when you have these colonies growing the CV space, the same way it is in the gut, they make things, they produce things. And what they produce can affect the epithelial cells and immune cells the same way it does in the gut. So we got very interested in digging into how live bacteria versus what they make affect the different epithelial barriers. And we assumed, wrongly so, that it would be the same. So again, we're back to cell permeability. This is live bacteria. Again, the dark bars here is Crispatus. The hash bars are GVAG, 10 to the 4th, 10 to the 5th, and 10 to the 6th of live bug. You can see here, 10 to the 6th really has a really big effect on cell permeability. So then we did the same thing with supernatant, right? Do you notice something's missing on this graph? So these are the supernatant from these bugs, and there's no increase from GVAG or from Crispatus. So it's only the live bugs that are really compromising the epithelial barrier. I want you to notice something here. I have a lot of stars on here for this not being significant. L. Crispatus supernatant made the barrier tighter. It fortified the epithelial barrier. So Lactobacillus live does nothing. GVAG live compromises the barrier, but the supernatants have a very different effect. This was honestly very completely surprising to us. We must have run this 10 times more than we needed to because I didn't believe the data at first. So then we went on and said, okay, are the supernatants and live bacteria really different? And what are they different by cell line, by the cervix or the vaginal epithelium? And are they different across the different bugs and the different cell lines between live and supernatant? So on the, left side of your screen is live bacteria. On the right side of your screen is supernatant. So here are the vaginal cells. So these are called VK2 cells. So go to the right-hand side of the screen. So here you've seen Crispatus. So blue means it lessened the cytokine response. The higher, the more orange or red, it increased it. So you can see here in the vaginal epithelial cells, L. Crispatus actually decreased baseline immune response, where you see GVAG was completely different. This is a supernatant. So then what we did is we did ecto, endocervix, and the vaginal cells in both the live bug, the live bacteria, and the supernatants. So if you looked at the ectocervix, right? So here's supernatant and here's live bacteria. So the supernatants, we see this really kind of profound effect that lactobacillus supernatants are having on the immune response almost in a protective way than the live bacteria. So really we only see a couple of cytokines from the live bacteria that are affected in the ectocervix. So now let's look at the vaginal VK2 cells from G. vaginalis. So in the live bacteria, as I showed you with the ability of live bacteria to break down the epithelial barrier and cause cell death, there is this humongous potent immune response where almost every cytokine on our Luminex array was significantly increased in response to live bacteria. From the GVAD supername, we do see an increase in the immune response, but it is much less potent than the live. So what this data told us, along with the one before it, is that there is a microbial specific, bacterial product specific, cell specific response in the CV space. So what have we learned to this time point? Microbes associated with health and disease have unique molecular effects on the CV epithelium. All CV epithelium is not the same. To be honest, this really shouldn't surprise us because they come from distinct embryological origins. But I think for a lot of work in this field for a number of decades, the CV space was thought as a continuous epithelium without unique properties, and that's just incorrect. But wait, I have a little bit more. So we really saw this very interesting thing between the microbial components and the different epithelial cells. So we wanted to ask on a much deeper molecular level what was happening. So we did this RNA sequencing where we put bacterial supernames or live bacteria on all three different cervical epithelial cell lines, RNA extracted, and did very routine RNA sequencing. So when you do these RNA sequencing, right, it gives you tons of data. And what you're really looking for are the different gene expression that is occurring from your different exposures. So because we had seen this difference in the supername and the live bacteria, we really want to know how they acted in each of these cell lines. And similar to, I love it when I'm surprised in the lab, and it's been 20 years and I'm still surprised. This data also surprised me. So the live lactobacillus on the ectocervical cells changed 21 genes. When we were more stringent with the twofold increasing using these barriers, there were 10. So for those of you who maybe do bench work or familiar with this, 10 genes to be changed is nothing. To me, I'm like, so did I even put lactobacillus on? It almost seems like it's an inert. So then we went to the lactobacillus supernames. Just so these are we let lacto grow. We took what it made in the media and we put it on the cells. So remember, 21 genes from live. We had 5,000 genes were differentially expressed from the supername, right? This really humongous difference where if you think about how lacto may work in the CV space to ensure health, to decrease the risk of HIV or to decrease STI acquisition, you kind of have to wonder, like, this is really different. These are profound molecular differences between live bug and its supername. So if you look at across now endocervical and vaginal cells, you see, so more were changed in the endocervical cells. And I would tell for anyone interested in epithelial biology, endocervical cells were the most potent immune responders out of all of the three cell lines and really quite different molecularly from the ecto cells. So this is what we saw from the live. And again, if you look at endocervical cells from live lactobacillus versus a supername, same thing in the vaginal, really profound molecular differences in how these cells are responding to supername versus the live bug. So what about GVAG? So you can see with live GVAG, consistent with what I just showed you for breaking down the epithelial barrier for the immune response, really potent molecular effects, 2,000 to 3,000 genes altered in each of the cell lines. I will tell you the gene pathways, which I show you, are different by cell line, but it's okay. So how does this compare to the supername? And what we see, again, pick your ectocervical cells, there's a much greater impact from live GVAG than from supernames, but we really still see pretty profound effects from GVAG supername as we do from live bug. So what are the gene pathways? I just told you there's these massive gene changes. What are they? So not surprisingly, so this is from ecto and endocervical cells. These are the gene pathways that were most significantly changed. And what I can tell you is not, as you might suspect, they are all related to cell adhesion and immune response. Though the immune pathways are different, I won't get into all of it right now, but they are different as far as it being whether it's inflammasome or whether it's bacterial signaling from TLR, IL-10. So there are different immune pathways between the ecto and endo, but all still very much immune related. And really begins to fill and ascribe mechanisms to this model that we talked about, that this immune response that's happening to having anaerobes in the CV space are really creating these diverse and potent immune effects in this biological niche. So I told you this weird finding with crispatus, right? That there's really not much but live, and then we see this kind of amazing molecular response to Lactobacillus crispatus supernatant. And if you remember when I showed you the epithelial permeability data, that crispatus appeared to fortify that barrier. So we were really, really surprised by the gene analysis, the gene pathway analysis for Lactobacillus crispatus. I'm assuming you can't see this, but what I will tell you, these top gene pathways are histone and epigenetic related. So it's not immune, it is not cell adhesion, it is all epigenetics. So Lactobacillus crispatus, on exposure to these epithelial cells, the supername, induces epigenetic modification in these epithelial cells. So before I go for that, I want you to think about how individuals with lactobacillus deficient communities are more likely to have worse serotypes for HPV, how they're more likely to have cervical cancer. Lactobacillus deficient communities are associated with HPV and cervical dysplasia. And if you think about that, lactobacillus rich communities may be able to epigenetically modify the susceptibility of epithelial cells to viruses or to proto-oncogenes. Maybe in your mind, think about how that may lead to reproductive health way beyond preterm birth. Okay, so what is exactly in the supername? So I just told you it's kind of a miracle thing. So what's in it? Well, there's a lot of things in supernames, but what we got really interested in is the idea of microvesicles and EVs, or extracellular vesicles. So we know that in eukaryotic cells, in mammalian cells, they can spit off these different things, right? They can have exosomes, they can have apoptotic bodies, or these microvesicles. So this has been known for a little bit of time now on eukaryotic cells, and the idea that EVs can traffic across the body in different biological niches to have an effect distant from the area of where the injury or something happened. But what is less known is that microbes, that prokaryotic cells can do the same thing. On the bottom of your screen is an example of EVs of these extracellular vesicles budding off a Pseudomonas. Right, so this has now been these idea that microbes, specifically, actually, most of the work's been done in E. coli and Pseudomonas, can bud off these vesicles that they can traffic and have a molecular effect is now known. And what's in those EVs can be anything from RNA to DNA molecules to virulence factors. And so we were really interested, is this true for what happens in the CV space? Are these common CV microbes able to make EVs? And so we found out, and they are. So this is the first time this has been reported. This is an NTA to look at the different size of the EVs, but basically here's our media where there's no EVs. So Lactobacillus crispatus, Mobiluncus, and Gardnerella vaginalis all are able to make EVs that are retrievable from this supername. So we really begin to think, well, these EVs must have cargo, that they must be doing something to induce these molecular effects. So they're there, but back to what I said before, we need to know whether they're actually functional. So the first thing we did, similar to what we did back with the microbes, the live bugs and the supername, is to ask whether they cause cell death. So again, on our bottom are ectocervical, our endocervical, and our vaginal epithelial cells. And then we had, these are EVs, right? So this NYC is the media that we grow up the bacteria in. So non-treated NYC, and then LC, Lactobacillus crispatus, Gardnerella, and Mobiluncus. And so really nothing. The EVs don't cause cell death, nothing seems to be happening. So that was good and bad. They don't cause cell death, which I guess teleologically maybe is good, but maybe they're just not functional. Maybe they're made and they bud off these microbes, but they don't have a functional effect. But they do. So what we found, so on what you're seeing here are 3 graphs of interleukin 8, which is one of a non-specific cytokine that we measure to see if there's an immune response from our epithelial cells. So as I told you, these endocervical cells are really immune potent and capable. So on your y-axis is the picograms per ml. So endocervical cells here go up to 6,000, vaginal epithelium to 2,000, and ectocervic to 4,000. And then what we did is we used different amount of EVs, no bacteria, no supername, just the EVs to see if they could induce IL-8. So what you can see here, and I'm draw your eye to the endocervical cells. So our media has no effect. Lactobacillus crispatus EVs have no effect on IL-8. Gardnerella, the increasing dosings have a pretty profound effect on IL-8, as do mobiluncus. And this held true for both endo, vaginal, and ectocervical cells. So these EVs that butt off these microbes, and if you think about that, EVs are easier to traffic. There's small little vesicles, could go to the, I don't know, the cervix, the upper reproductive tract, maybe even the uterus and beyond. They are able to induce from epithelial cells a significant immune response. Since IL-8 is nonspecific, and what I didn't show you is that our RNA-seq data showed from the supername there was a potent impact on the inflammasome and some antimicrobial properties. One of the other cytokines that we've been very interested in is CCL20 because it's a potent chemoattractant. So we looked at the EV's ability to increase CCL20. So as you can see here again, in ectocervical, endocervical, and vaginal cells, both Gardnerella vaginalis and mobiluncus EVs are able to induce a really potent chemoattractant from these underlying epithelial cells. So I had more on here. Good, I'm gonna go. So I don't know how many people are clinical for basic science, so we have a little bit of time. So I wanna go back to the bedside and show you some work that I think is relevant. So I told you in the beginning that we saw this race divergent colonization of microbial communities. One of, that maybe Alan could tell you about as well, one of my big soapboxes throughout my 20 years career is that we have excused or tried to explain racial disparities in obstetrical outcomes to genetics for way too long, that it is not biologically feasible that genetics is driving the increased rate of preterm birth, preeclampsia, or other adverse outcomes among Black individuals. And that is true for reproductive outcomes, including BV, STIs, et cetera. So we were really interested in why we found this. Why did we find a difference in the rate of non-lactobacillus dominated communities among Black individuals in our pregnant cohort? So thankfully, with one of my great colleagues and mentees, Heather Burris, who's an environmental epidemiologist, we were able within our 2000 cohort to geocode, to map where our individuals who participate in our study lived. So this is Philadelphia. This is Market Street, which is just an arbitrary designation. And what we found is if we took women from our cohort and just plotted them over Philadelphia, there was this area that had a 15% preterm birth rate and an area that had a 3% preterm birth rate. So if we call those a high and low risk area, traditionally in OBGYN, people would tell you, well, I know exactly the difference, right? It's lower socioeconomic, there's more tobacco use, et cetera. So we looked at the demographics between this high and low risk area. And again, remember this low risk is 13%, this is 14 to 15%. So completely refuting the biases that exist in our field, you will see that there's absolutely no difference in the demographics, these traditional demographics by how we judge risk between this high and low risk area in the individuals that were in our cohort. Even more importantly, we actually did this only with individuals who are self-reported as Black. So among Black individuals in our cohort in a high and risk, low risk area, there's no different about the demographics. So then what is driving the difference in the preterm birth rate? So I won't walk you through all the papers on the bottom, but we've actually found a number of things are associated with those areas. So maternal stress in the 15% area is associated with neighborhood violence. The local immune response, meaning vaginal beta defense and immune response in the CV space, is different in this area versus this area, and is associated with both neighborhood deprivation and maternal stress. And that violent crime and maternal stress are associated with both green space, and which are different within these two areas. We went a little deeper, and this again is Dr. Burris's work, to look at neighborhood deprivation. This is just a more detailed map of the one I just showed you. And this work is under review, where she showed that Black women live in census tracts with the highest deprivation, and that women with spontaneous preterm birth and the high-risk vaginal microbiota or a non-lactobacillus microbiota are more likely to live in those deprived tracts. For every standard deviation increase in neighborhood deprivation, women had a 30% higher odds of a high-risk microbiota. So what you begin to see is that where individuals live and their lived experiences and all the downstream effects of systemic racism affect microbial communities, which in turn affect reproductive health. So our new conceptual framework, along with what I showed you on the molecular level for understanding mechanistic underpinnings for adverse reproductive outcomes, is this. So our research program really stems from the idea that the microbial immune processes that drive health and disease in the CV space are influenced by all these different events, and that the reason we see such racial disparity in these events is driven from all the downstream effect of societal and systemic racism, and that if we are able to pick these pieces apart and look at the underlying mechanisms, that we will actually then be able to make a difference on understanding how to best prevent these outcomes for our patients. So in conclusion, the cervicovaginal space is a complex biological system, and hopefully I've convinced you of that. Microbial immune interactions appear to play a role in cervical remodeling and preterm birth. These molecular processes likely play a role in diverse reproductive outcomes, from infertility to STI to HIV acquisition. Microbes and microbial communities exert their effects through multiple molecular pathways, and these are bacterial specific, bacterial component specific, and epithelial cell specific. Understanding the complexity of this ecosystem is needed to advance our understanding of the pathobiology that drives adverse reproductive outcomes, and then can be leveraged, I hope, for precision-based therapeutics. This is my clinical and my lab team. Valerie Reese is my director of clinical operations, who made the motherhood and microbiome and the 2 cohorts we have ongoing for that. And this is my lab team, Lauren Anton, my senior scientist, and Andrea Joseph, my postdoc, and our students, from MD PhDs to PhDs to undergrads who have worked on these projects, and to my funding that makes all this work possible. Thank you for your time. Hopefully you are able to hear that. If you have any questions, I'm happy to take them.
Hey, Michal, this is just super exciting. Thank you so much. And I hope in the room there are questions, and if someone could go to the podium and pose those questions, that will be great. And for those on Zoom as well, I think we have good time for questions. So, Michal, let me what are some of the, before that, it's really exciting as well, as you know, because we are sort of breaking some new frontiers around social determinants of health, especially with the American Heart funding that includes PEN as well as others, because some of the work that we've done has been just within the institution, in the clinical facilities, but this is saying go outside. So for me, it's really informative to see how you're looking at things right at the genetic level, and how and then getting back to societal things. So what are some of the lines of investigation, especially with respect to interventions, that you are pursuing or will be pursuing as the next steps to this work?
So I think I'm far from alone in this. So if you guys are not familiar with, Caroline Mitchell at Mass General has been doing trials of vaginal microbial transplant. The idea with restoring a healthy microbiota. I think what we've learned from the C. diff, the fecal microbial transplant, that putting a single anaerobe in or putting something back in is not, there's a whole ecosystem that's created. So while I would love to think I could just give some lacto back, which people are trying, there are lacto trials going on in non-pregnant individuals across the country, across the world, actually. I don't know that it's going to be that simple. I think the better approach may be, as we figure out the EVs, the supername, and what the molecular path is, to actually increase that path, if you will, to up that path. But I don't know if that answers your question. We're doing a lot. I don't know that there's going to be any easy answer.
Thank you.
Jody asked, hi, Jody. Can you talk, you just asked the same questions. Can you talk about potential new interventions focused on intravaginal lacto to reestablish a healthy microbiome? Yeah, so I think some of this, and I have to credit Jacques Ravel with this. We talk a lot about whether the non-lacto microbiota is transgenerationally inherited, right? So as vaginal delivery and as you're colonized, that it's within communities that you maintain this microbiota, and that it's not always a reproductive risk. One of the things that, and I guess I had a little more time, but it gets so many detail, that we found is that women who have a high beta defensin, but a non-lactobacillus dominated microbiota, have a lower rate of preterm birth. So it's not just having lacto. If you have, quote, a good immune response, right? So what my guess is, I think some women learn to the cervicovaginal space tolerates the non-lacto and develops protective mechanisms, and so it's not an at-risk state. And I think more of that immune modulation may be a way to optimize reproductive health.
Great. I said, Dr. Berkowitz, yeah.
Yeah, thanks a lot, Alan. This was a phenomenal talk, really incredible. I wanted to just ask you from a more mechanistic question, the analysis of your exosomes, your microvesicles, did you do any metabolomic analysis of the, these organisms produce these short chain, medium chain fatty acids, and there's actually some really interesting data on receptors like bitter taste receptors that are present on vascular smooth muscle. I suspect they're present on some of these epithelial cells that may be driving some of this. In fact, there's some data that these bitter taste receptors may actually be the best tocolytics over, better than some of the beta, the beta agonists, etc.
So we've done a lot of metabolome work. So if you saw the kind of CV as a biological niche slide, so we've done a lot of metabolome work. We've done it in the human and in the vitro work. And it is really interesting. We don't see as much short chain fatty acids, though I still debate in the CV space if the metabolomic machinery is needed there for SFAs. We do see difference in lipids. We see different in short chain amino acids. And Kristen Gearson, who was a fellow faculty in my lab, has published some of this work. We have the in vitro data as well from all of the things that you're talking about. It gets really convoluted with the metabolome. And as it fluxes over time, I'm not as solid in that I believe the data. I will tell you we've done proteomics on the EVs, which is really interesting. The problem is there's not great databases to understand my prokaryotic proteomics, but there is tons of DNA and RNA cargo in the EVs. So I agree. So we know that TLR2 is acting in some of these and that vaginal lysine is being trafficked in the EVs for GVAG. I don't know what's happening in mobiluncus. We don't see that. And so we all are very much trying to figure out exactly what you're asking. I've not looked at the bitter taste yet to know, but yes, all of that.
Great. Lots of grants to write.
Yeah. There's another question here. Any potential other interventions target inflammatory markers at all on altering the microbiome? So there are some work. So we have found beta defensin 2. There's work, I just forgot where they're from. I apologize. Looking at beta defensin 3 in a mouse model as kind of an immunomodulator of the microbiota. Didn't talk about all the work that we've done in our mouse with the vaginal microbiome and creating a human vaginal microbiome. It's helpful and it's also not with the mouse, but I can talk about that another time. So there are, I think some of the inflammatory markers will. I think what we have to be really careful in the OBGYN world is thinking the immune response is simple. I think that's where we've gotten in trouble in the past. Brian asked, any thoughts on progesterone in the microbiome? Oh, yes, so many thoughts. And Jody probably knows this better than I do. There's this data, which I know goes back and forth, whether people believe it or not, with the Depo-Provera and HIV trials and the microbicide trials about the effect of progesterone in the CV space. We know clinically, right, when there's high levels of progesterone, if you think about postpartum and lactation, the CV space has less mucus, there's not estrogen, and there is almost a menopausal atrophic vaginal epitheliums like that. So to me, there is no way the sex steroid hormones are not influencing the microbiome. Speaking of writing grants, that's one of our other grants. I absolutely think estrogen and progesterone are modulating the EV space, the epithelial space, and how it allows certain microbes to flourish or not. You guys are probably familiar, right, that postmenopausally UTIs can be a big factor for women, recurrent UTIs. The best treatment for that is estrogen, right? In my mind, the reason topical estrogen is working is because it's changing the epithelial glycogen, changing the microbial communities, and therefore decreasing the risk of opportunistic infection in the bladder. So that's my thought on progesterone.
So, Michal, to use a bit of the time, I know some of the work you've been doing out there is really advocacy, really a strong advocacy around women's health. And would you care to just share with us some of the things you're doing with respect to recent developments around women's health, Roe versus Wade, etc.?
So you guys must have a hard time in Alabama. So Alan is correct. I'm a very strong reproductive justice advocate. I think, and as he knows, I've been pushing for the board not to have exams in Texas and all that. I think, to me, there's the obvious concern for Roe v. Wade is, for Dobbs, is that individuals do not have basic human rights. And that if you don't have the choice to make what you want to do with your own body, that speaks volumes to what other insults and harms will come to women. But I think it's more than that. I think, when I talk about, and again, if you follow me on Twitter, I've been pretty vocal on this. I think the problem is not just this moment in time. I think it's very much going to affect training. I think the ACGME trying to stand up booklets on how to learn abortion and DNCs is insane. I think that as we create generations in certain states that don't have training, those individuals, through no fault of their own, will go to other states that are not red states, but they will not be able to give the best care to women. I spoke for, I hope it's okay to get political, Josh Shapiro is running for governor in the state. He's very pro-choice. I spoke at a rally in favor of him. And the story I told that has affected me for my life was I was an intern at Cornell in New York City, and I got called to the ER, right? We had a pager that was called 1010. That was the intern. You got tortured constantly. Some of you may relate. And I got called down and I called, I said, what is it? They go, we need you now. Woman's bleeding out. I run down there. 16 weeks, she had broken her water on the way to the ER and was bleeding, and she was now in boots and she was hypotensive and she was dying. And her husband was in the room still, and it was a mess, and was yelling, this is a wanted pregnancy, but save my wife. Like, I don't remember, it was obviously a desired pregnancy. She was bleeding. And I remember immediately calling my third year, my chief, because obviously this was way above my pay level. And they started giving her blood, and my chief and third year were trained, and they rushed her to the OR and they did a DNC on a baby with a heartbeat, and they saved her life. And all I keep thinking is that if that happens in a few years in Texas or in Missouri or somewhere else, or even hear from someone who didn't get trained, that she'll die. And I don't know how that's conscionable. And I think it's really hard for you guys as residents to be in this position. And I just think we all need to fight.
Michal, thank you so much for the time, sharing this amazing science with us, and for your comments. I think the time is up, and I appreciate everyone who was able to join today. And you can find Dr. Elovitz if you look, and you can interact with her. But I just want to join others to say thank you very much. Yeah, and conclude. Great job. All right. That's it. Bye, everyone. Thank you, Michal.